Hello and welcome to Sun Biology class. Friends, today we shall discuss about cell-mediated immunity. Cell-mediated immunity is also known as cellular immunity. It is a form of immunity that is mediated by T lymphocytes. Cell-mediated immunity is the principal defense mechanism against intracellular microbes. This form of immunity is effective against intracellular microbes such as virus and some bacteria which survive and proliferate inside phagocytes and other host cells where they are inaccessible to the circulating antibodies. Cell-mediated immunity promotes the destruction of microbes residing in phagocytes and also promote the killing of infected cells in order to eliminate the reservoir of infection. Protective immunity against a particular microbe is usually induced by the response of the host to that microbe. Active immunity is a form of immunity which is induced by exposure of the host to the microbial antigen. This is known as active immunity because the immunized individual play an active role in responding to the antigen. Individuals and lymphocytes that have not encountered a particular antigen are said to be immunologically inexperienced or naive, whereas individuals, those who have experienced a particular microbial antigen and are protected from subsequent exposure, are said to be immune. There is another form of immunity called as passive immunity. Passive immunity is a form of immunity that can be conferred on an individual by transferring serum or lymphocytes from a specifically immunized individual. The recipient of such a transfer of either serum or lymphocytes become immune to that particular pathogen, though the individual has never been responded to that particular pathogen or been exposed to the microbial antigen. Passive immunization is a useful method for conferring immunity rapidly without having been to wait for an active immune response to develop. A physiologically important example of passive immunity is the transfer of maternal antibodies from mother to the fetus, where these antibodies provide protection against various microbial pathogens before they can develop their own system to produce antibodies. Passive immunization against toxin by the administration of antibodies from an immunized individual is a life-saving treatment for potentially lethal infections such as tetanus and snakebite. In fact, humoral immunity was originally defined as the type of immunity that could be transferred to a naive individual by the administration of antibody containing cell-free portion of blood, that is either serum or plasma, from an previously immunized individual. Similarly, passive immunity was defined as the form of immunity that can be transferred to a naive individual by the transfer of T lymphocytes from an previously immunized individual, but it cannot be transferred by the administration of plasma or serum. Antibodies are proteins that bind with toxins. Antigens are the substance that stimulate the production of antibodies. The modern definition of antigen includes substances that bind with specific lymphocyte receptors whether or not they stimulate the production of antibodies. An immunogen is a substance that stimulates the immune response. So friends, this is about cell-mediated immunity. In the next video, we shall discuss about the cardinal features of the immune response. For more updates, please subscribe my YouTube channel, Sam Biology Class. Thank you very much.